Okay, so my name is Marian Evangelio, and I came from the Autonoma University of Madrid. Me with my group, we are part of the SP1, which means that we are mapping cortical and subcortical connections in the mouse brain. And more precisely, I'm working in this center here in the thalamus, in thalamocortical connections, and more precisely in the visual part of the thalamus, which is connecting to all the visual cortices, primary and secondary. So why is important the thalamus? Because it's an entry route of sensory information. It's a feedback to the cortex to control the output signals. It's also a control of the cortical arousal and regulates the communication between particular sets of cortical areas. But more important is that it's crucial for modeling normal and diseased brain functioning. So what we do in the lab, we operate the mouse, and with a micropipette, we put in the brain, in the thalamus, a color molecule that can be an amine or a fluorescent tracer or a virus. And then after the survival period, we remove the brain and we slice it we cut it in sagittal slices, as you can see here. And then we dye these slices and we can see the cortical projections, the cortical terminal field of the axons. And so we can also reconstruct the projection of these neurons. But for that, what we have developed is this map of here, which is a map, or it's a flat map of the cortical surface of one hemisphere. So here would be the frontal pole, which is here between the eyes, and here the occipital pole. Here is bregma, which is the medial part of the hemisphere, and here would be the lateral part. And we use this map to represent the axonal terminal fields in the cortex. So here there's a magnification of V1 and primary visual cortex and secondary visual cortex with the retinotopical map, because it's the main target of the neurons that I am studying. So our approach, first of all, is going to be in a population level, which means that we are going to stain a group of neurons between 8 and 10, and then we're going to go to a single cell level approach. So first of all, we were interested in do a retrograde study, because what we wanted to do is put a retrograde tracer in the cortex, and then the terminal fields are going to eat this substance, and it's going to feel all the way backwards to the soma. So later we can see here the somas illuminated for the neurons that are projecting to this region of the cortex. Here you can see the injections that we did and the position, the place of the somas. Then we did an anterograde traction study, and we did really small injections in all the nuclei in the thalamus that are implicated in the visual system. And here you can see from this neuron, you can see the cortical terminal field of the axons that are coming from here. Here you can see a magnification of the axons in the cortex. And at the end, what we did was a single cell study, which means that we used DNA plasmids and from adeno-associated virus, and also RNA from this virus. And what we did is electroporated it, which means that we open pores in the membrane and we put inside the DNA or RNA. And then these neurons are going to take this substance as her own or his own, and it's going to fill the whole neuron. And then after 5 weeks, what we can see is illuminated the soma here, because it has a GFP, a fluorescent molecule. And then when we dye it, we can, with neurolucida, we are able to reconstruct the whole neuron and the whole terminal field. And at the end, what we wanted to do, we didn't want only to have the 3D reconstruction, but also do a quantitative analysis. So we use neurolucida and stereology, these two techniques, to find out the total axon length in the cortical in every cortical area that are projecting these neurons, and also the number of varicosities or synaptic buttons that are putting these neurons in every layer of the cortex. And if you have more questions later, you can ask me in the poster. Thank you very much.